This is a short description of our VR lesson on the nature of icosahedral viruses and how they attain their size. We cover viral capsid function and symmetry and explain triangulation number, quasi-equivalence, and Caspar-Klug theory through hands-on interactions. First, we examine the small satellite tobacco necrosis virus to show how it is composed of simple protein building blocks that are assembled to encase the virus's genetic material, which is inside. Next, we look at the nature of icosahedral symmetry. The virus uses to build its capsid by using many copies of the same building block. These two geometric shapes have identical icosahedral symmetry elements. One is made of 20 equilateral triangle faces, each containing 3 identical subunits. The other is made of 12 pentagonal faces containing 5 identical subunits. Thus both contain 60 identical subunits. Their symmetry consists of 3-fold, 5-fold, and 2-fold symmetry axes. We can take a single kite-shaped subunit and create a pentagonal face using a 5-fold axis. Using a diagram on the table, we can place 3 pentagonal tiles around the 3-fold axis on the vertices of one icosahedral triangular face. We can then build 12 pentagons to form a complete capsid structure. Since all subunits are identical symmetry environments, this has a triangulation number of 1, or T1. In order to make larger capsid structures, we must add more subunits and go beyond the 60 subunits in identical symmetrical positions. This exercise uses hexagonal tiles that may have 2-fold, 3-fold, or no internal symmetry, as shown by its color. Here we place a tile that has 6 different colors indicating no symmetry. The resulting capsid structure, with its pentagons and hexagons, has subunits of 7 different colors, which indicates 7 different symmetry environments and is a T7 structure. Next, move to our virus dispensary, which can produce a large variety of actual virus capsid structures. As we page through the viruses, we see a variety of icosahedral capsids with different protein building blocks, sizes, and T numbers. We can manipulate them, examine them, probe them with our symmetry tacks, and even explode them into their subunit components, finding subassemblies which may have local or quasi-symmetry between the identical protein components, but do not lie on the icosahedral symmetry axes. We then go to the T-number generator board, which is based upon the mathematical Caspar-Klug analysis of icosahedral virus capsids. Here we can create any size regular capsid structure by manipulating the vertices of the icosahedral triangle on a hexagonal mesh, selecting the placement of pentagonal vertices, which results in selected T-number particle. We can select the type of subunit shape, colored or drawable, automatically populate the triangle, and build the capsid. If a drawable subunit is selected, one can draw on the tile and see the result on the entire capsid. Finally, we can take our newly built capsid structure to our drawing board, which enables one to be creative in designing their own unique icosahedral capsid structure. We can draw and fill each individual tile in the icosahedral asymmetric unit and see how it develops on the entire capsid. The diameter of the drawing pen can be selected and changed, as well as the color and the opacity of the pen and the fill. One challenge is to produce a capsid which has quasi-symmetry elements with local colors or drawn shapes. Here we color a true threefold tile and then an asymmetric tile with quasi-threefold coloring. When finished, the symmetry tacks can be used to delineate icosahedral triangle on the particle. If the tacks are placed correctly, fireworks ensue to indicate success.